Hello, everyone. We are delighted to have as our seminar speaker today, Dr. Archana Unnikrishnan. She is an assistant professor in the Department of Biochemistry and Molecular Biology at the University of Oklahoma Health Sciences Center. Dr. Unnikrishnan's early work focused on the impact of dietary factors on genomic stability and carcinogenesis. She then transitioned her research direction to studying aging, a topic that is dear to all of us here at the HNRCA. As part of her aging research portfolio, she has studied the effects of dietary restriction on lifespan and health span, including insulin sensitivity. Using different model systems and interventions, she has examined epigenomic and transcriptomic mechanisms of longevity and the impact of dietary interventions on anti-aging pathways. In studying the various insulin response tissues, her focus has largely been on the intestine since it's important for both insulin sensitivity and as a potential site for what we now know as inflammaging. Dr. Unnikrishnan's current studies also characterize the effect of GLP-1 receptor agonists in the improvement of intestinal stem cell function. Her talk today is titled, Can Dietary Restriction Improve Intestinal Aging? And before I turn the microphone over to Dr. Unnikrishnan, I would like to request our seminar attendees to please use the Q&A box to post your questions, and you can do this as and when you have them ready. Thank you all for joining, and we look forward to hearing from you, Dr. Unnikrishnan.
Thank you, Dr. Das, for the introduction. And thanks to each and every member of the HNRCA for giving me this opportunity to present my work. And today, as my title says, I'm going to talk to you about what we know about dietary restriction and its effect on intestinal aging from animal studies. To start with a little bit of introduction, calorie restriction or dietary restriction is not a new concept. It has been around since the 1930s. The first study on calorie restriction actually came out in 1930s from Clive McKay's lab, where he was studying the effect of food restriction on growth retardation. So what he had done is he had exposed his model, which was rats at that time, to food restriction, I guess about 30% restriction right after weaning or 2 weeks post-weaning, and he followed them. As I said, his intention was to retard the growth, and he used food restriction as a way to achieve that. And he saw that the food restriction really did get the growth retardation that he was expecting, and at the same time, the rats which were on food restriction, both right after weaning or 2 weeks post weaning, both the cohorts showed increased lifespan. So this is not a really new concept. So we are almost getting to celebrate the 100 years of this first research paper that came out. So how do we do this food restriction or food intake studies in these rodents? There are several ways of doing it. The first one is the classic way, which we call it the 40% Maseru diet, where you are reducing the amount of food that is fed to the rodents who are on ad libitum feeding. Now, here I want to take a moment to differentiate between calorie restriction and dietary restriction. Calorie restriction would be just reducing the calories without any changes in the micronutrients, whereas dietary restriction is reducing the entire diet in total without adjusting for the vitamins or minerals. But then studies have been done so far to show that there is no nutritional deficiencies because of that. Then the other ways of reducing or doing food restriction studies is by intermittent fasting, where you can do alternate day fasting or time restricted feeding. Or other ways are low protein diet, essential low restriction in essential amino acid such as methionine. And right now, there are also studies where they are limiting branched chain amino acids and looking at the effects of lifespan. And finally, fasting mimicking diet or a periodic low calorie, low protein diet. Mostly this is done by giving a 10% restricted diet for about 3 days, twice in a month. So there are several ways of imparting for dietary restriction in rodents. And my study especially follows the Masero diet, and we don't do the calorie restriction, we do the diet restriction. So this lifespan data that I am showing you is with dietary restriction. And as you can see, right from invertebrates, yeast to flies to rodent models to dogs to monkeys, 30 to 40% dietary restriction or calorie restriction has been shown to increase lifespan across the species. And at this point, dietary restriction is considered to be the most robust intervention that has been consistently shown to have an impact on lifespan and even health span in the rodent model. So this concept was until 2010 when a study came out which showed that dietary restriction might not be, or 30 to 40 percent dietary restriction might not be a good restriction model for all genotypes. So in that particular study, what they had done is they had about 48 to 49 different strains or recombinant inbred strains of mice, both male and female, and they exposed them all to 40% dietary restriction and followed their lifespan. And they saw that about a third of them responded to dietary restriction, as we have seen before. A third of them did not show any response at all. And a third of them actually had a negative, that is reduced lifespan with 40% dietary restriction. So when I moved to Oklahoma in 2010, I collaborated with Arlen Richardson, and we undertook this idea that with all genotypes might not be able to adjust to 40% dietary restriction. So, but maybe if we reduce the dose of that to a 20 or a 10%, maybe all of these strains which did not show a response or showed negative response will have a better lifespan outcome. So we had about 8 strains of mice which showed varied response with 40% dietary restriction, and we had them on 40, 20, and 10% restriction. So now this data that I'm showing you here is C57 black 6 mice, which has consistently been shown to have improved lifespan with dietary restriction. The blue curve here is that of the ad libitum. The yellow one is 10%. The orange is the 20, and the red is the 40%. And as you can see here, the 20 and the 40% significantly increase the lifespan, the 40 being the best. And then we used this recombinant strain of mice, stage 115, which out of that study in 2010 actually showed that they live only for 3 months with 40% dietary restriction. So to our surprise and shock, they lived beyond 3 years. So, and that too, even with 40% dietary restrictions. So they showed improvement with 10%, 20%, and the 40% again was the best. But out of the 8 strains that we had used, there were a couple of them which did not respond very well to 40%, but they did better with the 10 and the 20. So the take home that we got from here was, one is 40% might not be good for all strains, but maybe if you adjust the dosage based on the genotype, there might be a beneficial effect. And the other thing that we learned was this particular strain, which lived shorter only for 3 months in the previous study, and in our hands they lived beyond 3 years with 40% restriction, was because the number of animals they had for the lifespan study was very, very low. So that's another key thing when you're doing lifespan studies with rodents, especially with all these interventions. You need to have a large cohort to actually see the effect. And this study came out in 2024 from Jackson Laboratories, where they actually compared intermittent fasting and the regular dietary restriction, 40% dietary restriction, but they used a diversity cross, outbred cross mice to look at the effect of these diet restrictions on lifespan. So they had 5 groups of mice, again, all female outbred mice, ad libitum group, and a group which had 1 day intermittent fasting, the third group, which had 2 days of intermittent fasting, then a cohort which was on 20% diet restriction, and the fifth cohort, which was on 40% diet restriction. So, and when they looked at, and the restrictions was started at, or the feeding regimen was started at 6 months of age. And when they looked at the lifespan, as you can see here, the 40% dietary restriction was the one which showed the maximum impact. But the 20% also had significantly increased the lifespan, median and the maximal lifespan. And the intermittent fasting for 2 days also had a significant increase in lifespan. So here, I'm showing you the 50 and the 90% lifespan data on for each of these groups. And as you can see, the percentage increase from the ad libitum that is given here. And you can see that 40% increased both the median and the maximum lifespan. So did the 20, whereas the intermittent fasting only increased the 2-day intermittent fasting increased only the maximum lifespan. So from this up to now, it's at least in the rodent model, the 30 to 40% dietary restriction seems to be working the best. So as I was working with this 8 strains of mice, and this was in a naive way that I joined the aging research at that time, and I didn't realize how so much work is involved in doing calorie restriction studies that you have to go down there to feed them every day, pair feed and all of that. And then mice, which were supposed to live only for 3 months, are now living for 3 years. So it was a big undertaking. So as we were working on that, my thought process was, is there a way we can do a short term, not an intermittent fasting or fasting mimicking diet or anything, but do the regular macro diet of dietary restriction, but only do it for a short period of time and have an impact on it. So I started digging into literature. And then I found this paper from 1985, again from Massero's group, where they had done work on rats. What they had done in this study is they started 40% dietary restriction right after weaning and allowed these rats to be on that restricted diet for up to 6 months of age. After which they were switched out to ad libitum feeding, and they were followed for their lifespan. And as you can see here, there was a significant increase, about a 13% increase in median and significant increase in maximum mean and maximum lifespan as well. So which means that, okay, the short-term dietary restriction seems to be working. If it has an impact on lifespan, it could be having impact on other factors as well. So this could be a potential area that can be pursued. So the next question was, what would be the physiological effect that we would go after? What would be the health span effect that we would actually measure? So if you look at dietary restriction or calorie restriction, at least from the rodent models, we know that it has an impact on various age-related diseases, such as neurodegenerative disease, cancer, cardiovascular disease, diabetes, kidney disease, and even autoimmune disorders. So we chose for our short-term studies to look at insulin sensitivity and glucose tolerance and see if short-term dietary restriction has an impact on it. So in this particular study, what we did this on young animals, not on aged. Young animal, we took young adults and we put them on 40% dietary restriction for about 4 months. And then after that, we switched them out to ad libitum feeding for about 2 to 3 months. So the data that I'm showing here for each of this graph, and we did the body composition and we did the glucose tolerance test on these cohort of mice. So the data that I'm showing you for each of those graphs, the first white that you see is the ad libitum. The red is the 40% restricted. And this is a cohort where they were still on restriction. And to the right, what you see here is the ad libitum and the cohort of mice which was switched out to ad libitum feeding after 4 months of restriction. So as you can see, there was decrease in body weight after 4 months of restriction. And once they were switched out to ad libitum feeding, the body weight came back up. But when we looked at the fat composition, it was significantly reduced after the 4 months of restriction. And even when we switched them out to ad libitum feeding, the total fat content seemed to be reduced. So when we looked at glucose tolerance test, within the first 4 months of dietary restriction, there was a significant improvement in glucose tolerance. And when we switched them out, we again saw a significant increase or significantly improved glucose tolerance. It is not as good as what it was when it was restricted, but nevertheless, it was improved glucose tolerance even when they were switched out to ad libitum feeding. So this gave us an idea that, okay, short-term dietary restriction seems to be having some kind of metabolic memory. And then this type of study was repeated by others as well, where we had done only 2 to 3 months after switching out to ad libitum, but some studies have gone up to 9 to 10 months of ad libitum and looked at insulin sensitivity, and they seem to be improved. So short-term dietary restriction seems to be having a metabolic memory here. So we wanted to test this on a different model. So we used a pre-weaning model, which is a crowded litter model, where the food restriction is done actually during the lactation period. So this is a model which was created by Rich Miller's group at University of Michigan. What they do is, once the pups are born, they are cross-fostered. For example, C57 black 6 mice, their average litter size is 6. For litter expansion, what we do is we cross-foster them, move the animals to another mother, and we increase the litter size. So a mother will have 12 pups, so 100% increase in litter expansion. So our control group had 6 pups, whereas our experimental group had 12 pups. So 100% increase in litter size. So Rich Miller's group, as I said, had already done this. They had done it on HET3 model, and they actually saw that there is increase in lifespan. About 18% increase in lifespan was observed in the expanded group, the animals which were part of the litter expansion. So we used the same model, but we used a C57 black 6 strain of mice, and we followed them for about 15 months of age. So the light blue graph bar you see here is that of the control ones, where we had 6 pups in 1 liter, and then the dark blue ones had 12 pups per liter. And we followed their body weight longitudinally for 15 months. And as you can see, it was significantly reduced right from the beginning, from 1 month of age to 15. And when we looked at the body composition at 15 months of age, their total fat level was also reduced. So we did similar to what we had done with our post-weaning model. We did glucose tolerance, and we did insulin tolerance test as well. We did this again longitudinally at 5 months, then at 9 months. So with glucose tolerance, we did not see much change at 5 months, but 9 months, they were actually improved insulin glucose tolerance. And when we did the insulin tolerance test, again, at 5 months, we saw there was a significantly improved insulin tolerance, and 9 months, it was even more robust. Now, the key thing is, the restriction, the food restriction or the competition for food was created only during the lactation period. Once they were weaned out to regular chow diet, and they were fed ad libitum. So there was no food restriction of any sort once they were weaned on. And then as you can see, like the insulin tolerance and glucose tolerance, everything seemed to be improving with age at 9 months. And then their adiposity and their body weight, everything remained improved even at 15 months of life. So food restriction in both these models, post-weaning and pre-weaning models, seem to be leaving behind a metabolic memory. So we wanted to understand what is the molecular mechanism underlying this metabolic memory. So for this, we used the same short-term dietary restriction model I mentioned before, where they were restricted for about 4 months, and then they were switched out to ad libitum feeding, and for about 2 to 3 months. And we harvested tissue from each of this cohort, and we decided to we wanted to study the mechanism across the biological spectrum, from the methylome down to the metabolome. So we decided to start with the transcriptome. So, and we focused on insulin responsive tissues, such as intestine, liver, hypothalamus, hippocampus, and there were a few others. So now what I'm showing you here is the blue represents genes which were increased with dietary restriction, and red represents genes which were reduced in expression with dietary restriction. And within each of this pie chart, the darker color represents genes that changed, and the lighter color represents the number of genes that persisted, that even when they were switched out to ad libitum feeding, the number of genes which changed with dietary restriction and persisted even when switched out to ad libitum feeding. So as you can see, there are a large number of genes that changed with that 4 months of restriction, and then quite a bit of genes that actually persisted even when they were switched out to ad libitum feeding. So now, what is responsible for this transcriptomic change or the persistence of expression pattern that we see here? So to look at that, rather than spreading ourselves thin with all of these tissues, we decided to focus on one particular tissue, which is the intestine. So, and there are other reasons, just as what Dr. Das had mentioned early on, good reasons to look at intestine, in the sense that intestine does undergo changes with the age. It is a center for inflammaging, and leaky gut is responsible for various gut liver and gut brain axis dysfunction, and with aging, intestinal aging is a risk factor for cancer, inflammatory bowel disorders, colitis, and Crohn's. And at least in animal models, dietary restriction seems to have an impact on each of these intestinal aging effects. So that is also one of the rationale why we decided to stick with intestine. And then previously from our lab, this was a study which we had actually done in collaboration with Dr. Promislow's group there. And we looked at the effect of age and dietary restriction on intestinal microbiota. So here what we did was we put young and old animals on dietary restriction, and we took the fecal colon feces as well as some of the fecal matter from the small intestine as well, and we looked at we analyzed them for the microbiome. So when you do the PCA analysis, the young ad libitum microbiome is right here, and this is the old ad libitum. So you can see the difference, how far apart they are. So when we put the young animals, young adults on calorie restriction, their microbiome clustered right here, closer to the young ad libitum. So there's some overlap there. So, but when we look at the old dietary restricted microbiome, they're right here. They are not overlapping with the young, but at least they are clustering closer to the young and far away from the ad libitum group. So which again showed us that, okay, probably calorie restriction seems to be having an impact on the intestinal microbiota, and they are trying to make it look more of a young phenotype. And when we looked at several species of bacteria, and we saw that several of the pathogenic bacteria which goes up with aging was reduced with dietary restriction. And when we did the ratio for bacteroidetes to firmicutes, now bacteroidetes are considered to be the good bacteria, which is good for insulin sensitivity and reducing inflammation, and firmicutes does the opposite. You can see the young animals both have the ad libitum as well as the DR group kind of have a similar response to the ratio. But when you look at the old ad libitum had only firmicutes. We couldn't find any significant amount of bacteroidetes in there. Whereas in the old animals which were exposed to calorie restriction or diet restriction, you can see that bacteroidetes level is going up. So dietary restriction seems to be having an impact on the intestine. And based on all of these reasons, we decided to stick with the intestine to study the short-term dietary restriction effects. So going back to the previous study where we had done the transcriptomic analysis, we took those gene set and we did pathway analysis, and what we saw, so this is ad libitum, this is dietary restriction, and this is the dietary restriction switched out to ad libitum. And as you can see, some of the top hits were genes which were involved in glycolysis and gluconeogenesis. And then we also have some from the Wnt signaling pathway, which I'll circle back to the importance of that pathway in a few slides. So there's some indication that the genes that seems to be changing with dietary restriction and are persisting even when switched out to ad libitum feeding is responsible for glycolysis and gluconeogenesis, which in turn can be having an effect on insulin sensitivity. So the next question was, we had a functional readout now, and we did see a transcriptomic memory change that happened. So we wanted to understand what is controlling this gene expression changes. So the obvious place to look at was epigenetics. And based on my previous work during my doctoral period, I decided to look at DNA methylation. And DNA methylation, as you all may know, it's the cytosine which gets methylated at the at the fifth position. And then you have, this is the methylation mark, and you also have the 5-hydroxymethyl cytosine, which is the demethylation mark. So these two modifications of the cytosines are read out as DNA methylation. So why DNA methylation? This has been identified long ago, 1948. It can, whatever change you see are long lasting, but at the same time, they can be dynamic. And mostly the methylation occurs in Cs followed by Gs, and the CpG is have a higher density in the gene promoters and then are said to have an impact on gene expression. And the classic view of this is that if you have CpGs in the promoter regions and if they are left unmethylated, then the gene gets transcribed. And if the CpG sites are methylated, then the genes are silenced. So this is the classic view of how DNA methylation regulates gene expression. So how do we do this assay? We do bisulfite treatment on the DNA that we get from these intestinal mucosa. And this bisulfite treatment converts the cytosine into uracil if they are unmethylated. If they are methylated, then they are not converted. They remain as such. So this is a sequence. You have a cytosine, you have a methyl C and a hydroxymethyl C. On bisulfite treatment, the unmethylated Cs gets converted into uracil, whereas the methylated Cs and hydroxymethylated Cs remain as C. And when they undergo PCR, the uracil becomes a T, whereas these two Cs, which are methylated, remain as C. So now when we do this heavy sequencing, next generation sequencing, and we compare them to the original sequence of the DNA, you can see that if the purple ones here are the ones which turned into T, and the reds are the ones which remained as C. So which, if you see a lot of red, then that means they are methylated. So in this particular site, if you can see, there are 6 T's and 4 C's, so there's about 40% methylation. So this is how we measure that for each of those CpG sites. And then we have a count on what is the percentage of methylation at each of those CpG site for your entire region that you have analyzed. So based on this, we did several candidate genes. The one gene that I'm going to show you today is neurotensin 1. Now, this is a gene which, again, has two important role in intestine. It plays a role in insulin sensitivity, plays a role in glucose metabolism, and at the same time, they're also involved in stem cell function. So when we look at the expression of this gene, which is from the RNA-seq data, with 40% dietary restriction, the gene expression goes up. And when they are switched out to ad libitum feeding, the expression remains upregulated and it doesn't go down. So when we looked at the methylation as of the promoter region, the 500 to 600 base pair region upstream of the transcription start site of this particular gene had only 3 CG sites. And we looked at the methylation level of these 3 CG sites. This is site 1, site 2, and site 3. And for the 3 conditions that we had. And as you can see with dietary restriction, the levels go down and it stays down when they are switched out to ad libitum feeding. So based on the classic view of how DNA methylation regulates gene expression, there's a reduction in methylation here, which could be responsible for the increase in gene expression that we see here. So again, this is not a cause and effect, but we see an association between the methylation changes that is happening with short-term dietary restriction and the gene expression. So now here, I want to take a moment to explain why we switched from here to intestinal stem cell. So until now, the work that we had done with the transcriptome and methylation and everything was on the intestinal mucosa. So the intestinal mucosa is like when you open the intestine and you open it and you slough off the top area, and that's your mucosa layer. And all our study was basically done on that. So when you look at the structure or the morphology of an intestine, you have a crypt base at the bottom here. And then you have this huge structures, finger-like projections, structures called as villus. Now, this finger-like projections, these are the structures of the intestine, which actually has the differentiated cell types that make up the intestine, mainly the absorptive cells or the enterocytes, and you have the other tough cells and goblet cells, which produces the mucus layer and all of that. So this, and the crypt base is the one which houses the stem cells, and it also has its own feeder cells down there, which are called as Paneth cells. Now, when in our previous study, when we were doing the transcriptome and methylome, we were looking at this layer. This is the region that we were looking at for the expression and the methylation pattern. So now when you look at how intestinal physiology, these top layer or the differentiated cells that you find in the villus, they get sloughed off every 3 to 5 days. So once they are sloughed off, the intestinal stem cells at the crypt, they have to replicate, rejuvenate, and replenish the enterocytes or the goblet cells or the tough cells, which are removed off after every 3 to 5 days. So which means that this intestinal stem cell have to be highly active and highly proliferative and should have the differentiating capacity to regenerate the entire intestine almost every 3rd or 5th day. Now, so if we are seeing changes in methylation in some of the cells up here, that means that methylation changes has to be happening down here. And as I was mentioning earlier, when we looked at the pathway analysis, we did see that there were changes in Wnt signaling, which basically is a pathway which is responsible for intestinal stem cell proliferation and differentiation. So from now on, we decided, okay, to understand what is going on with this memory effect with short-term dietary restriction, we decided to look at what happens to the intestinal stem cells. So these intestinal stem cells can be isolated from the mouse intestine and can be put in culture. This is an illustration showing starting from a single stem cell. And from the single stem cell, you can grow them in culture. Up to 5th day, they are proliferating. And from then on, they start differentiating and start budding out. And by day 19, you have a organoid or enteroid, as we call it, with a lot of these buds, each of this bud representing one crypt of the intestine. And this is a fully functional intestine. You can actually do permeability assays and all of that in this enteroid. And it is a surrogate way of also testing how functional or how active your intestinal stem cells are. So using this system, we wanted to address, does ISE function decline with age? Does short-term dietary restriction have an impact on this ISE function? And does the short-term dietary restriction impact the transcriptome of the ISEs? So the data that I'm showing you here was published last year. And here what we are showing is intestinal stem cells isolated from young and old ad libitum mouse, and they were grown in culture. and allowed to form enteroids. And as you can see, the young ones had a large number of enteroids and enteroids which had these buds, which is a fully functional enteroid. And this is day 15 in culture. And with the old, we see a much less number of enteroids compared to the young. And not only do we see a decreased number of enteroids, even the enteroids which have this multiple buds are very much reduced in the old condition. So this is again the same data. I mean, the previous slide that I was showing you was in a 4x magnification. Now this is in 20x magnification, and this is day 3, 5, and 15. This is from young ad libitum. You get a good enteroid by day 15. And then this is a young adult exposed to 4 months of dietary restriction. As you can see, it improved the enteroid formation even better than the ones in the ad libitum. And this is the old ad libitum, where by 5th day, they're kind of fizzling out. They don't do much. They don't bud anymore. And this is, so here I want to take a moment to say that this is the first time that we were doing any kind of short-term dietary restriction in old animal. So we took 21-month-old mice and we put them on dietary restriction for 4 months, and about 25 months of age, we sacked them, isolated the ISEs, and we looked at it. And then, as you can see here, nice enteroids are formed from those ISEs. So short-term dietary restriction seems to be improving intestinal stem cell function, at least by way of enteroid formation, not only in the young, but also in the old animals. So even if you can start the dietary restriction in old animals and have a late life beneficial effect out of the short-term dietary restriction. So this is 4x magnification of the same. As you can see here, this is ad-lib young, ad-lib old, DR young, and DR old. The DR, both in the young and the old, they seem to be improving the enteroid formation quite a bit. So then we took those enteroids and we looked at gene expression pattern of those enteroids. And what we saw was the orange and the red here represents young DR and old DR, whereas the green in these case represents the young DR and the old young ad-lib and old ad-lib. And as you can see, dietary restriction seems to be improving regeneration markers, which is Wnt gene and TERT gene, and it seems to be reducing the quiescence marker and also reducing the cell cycle RS marker. So basically, they seem to be rejuvenating or keeping the stem cells poised in such a way that they can undergo proliferation and differentiation. So we went back to the freshly isolated stem cells and we looked at the transcriptomic analysis on that. And when we did that, the top 38 genes that we found to change with age and were reversed with and the age-related change reversed with short-term dietary restriction, when we did a pathway analysis on those, we found that most of those genes were involved in cell-cell adhesion, or they were receptors or cellular markers which are responsible for bringing in other cell types closer to them, which could be promoting their proliferation and differentiation. So again, we went back to, we had this idea, the concept of methylation could be playing a role in gene expression analysis of these genes which are changing with short-term dietary restriction. So we went back again to look at methylation changes in them. So, but before we actually looked at the methylation changes, we wanted to see if there is a functional memory here as well. So earlier we saw with glucose tolerance and insulin tolerance, there was a metabolic memory. With gene expression changes, there seemed to be a memory effect. But with stem cell function, is there a functional memory effect as well? And if there is a memory effect, then maybe looking at DNA methylation would be the best way to go about. So this is the same images that I showed you earlier. This is the young ad-lib. And this is the old ad lib, which did not form good enteroids by day 15. And this is the old ad lib, which were on 40% dietary restriction. As you can see, they form good enteroids. And now what we did was we took a 25-month-old, which were on dietary 40% dietary restriction for 4 months, and we switched them out to ad libitum feeding, and we waited until they were 30 months old. And at that age, we isolated the stem cells and we looked at the enteroid formation. As you can see, they formed beautiful enteroids. So there seems to be that 40% dietary restriction, even when started in old mice and kept only for 4 months, when you switch them out to ad libitum, there seems to be some sort of functional memory that is left behind in those intestinal stem cells. So now this gives us a good rationale for looking at DNA methylation. And we did several genes, but I'm just showing you a couple of them. The first one is SLC28A3, which is a gene which is responsible for purine and pyrimidine transport, which again plays a key role in proliferation and differentiation. And as you can see here in this graph, this is young adlib. Dietary restriction. With dietary restriction in the young, it goes up. With age, it goes down, and then it comes back up with dietary restriction in the old. So when we look at the methylation of this particular gene, we did look at the methylation in the promoter region, and we did see some changes there as well, which are associated to gene expression levels. But the one that I'm showing you here is the methylation change that we found in the gene body of the gene. And research these days show methylation changes that you would find in the gene body is more associated to gene expression than the ones that you find in the promoter region. So when we look at this, there were about 3 CG sites, and when you look at the methylation of each of those CG sites, it is reduced with short-term dietary restriction both in the young as well as in the old, which kind of associates very nicely with the gene expression change that we see for SLC28A3. Similarly, we followed a Ly9. This is a gene which is a cell surface marker, which is involved in bringing in immune cells, and then they are responsible for insulin. I mean, they are involved in insulin sensitivity and also regulating inflammation. And with the age, their expression level seems to be going up and dietary restriction seems to be reducing it. And when we looked at the methylation of the 3 CG sites, so just to back up a little bit, when we looked at the DNA methylation, I'm just showing you representative regions, but we did look at 4,000 base pair region upstream of the transcription start site and downstream of transcription start site. And we did see several regions of differentially methylated regions, which associates very well to the methylation change, to the expression changes of these genes. So I'm just showing you representative regions here. And as you can see here, the methylation levels from with age as well as with the dietary restriction in the young and the old associates very well with the expression of Ly9 genes. So again, this is association, a correlation, not a cause and effect. But at least it tells us that methylation could be playing a role in regulating the expression of these genes. So in summary, what do we know is short-term dietary restriction can have a memory effect. It improves insulin sensitivity. It seems to be having an impact on the transcriptome of the intestinal mucosa. Apart from that, short-term dietary restriction seems to be improving ISE function in aged mice. You can start them even at an older age and get a beneficial impact out of it. They seems to be leaving behind a functional memory. And the short-term dietary restriction reverses the age-related transcriptional changes that you see in the old mice. And when we look at several of these gene expression changes, they seem to be associating or correlating with DNA methylation changes. So this is not the end of it. This is just the beginning of the study. As I said, we want to go across the biological spectrum, right from the methylome down to the metabolome. And even when we look at DNA methylation, there's a lot more work that needs to be done. So some of the current studies that is ongoing in my lab right now is these freshly isolated ISCs. We took them and we have done single cell transcriptomics on them, and we find within that intestinal stem cell population there are 4 different clusters, and each of those clusters seem to be, so the white is the young ad lib, black is the old ad libitum, and the red is the um, old animals which were exposed to dietary restriction. And as you can see that age seems to be having a different impact in each of these clusters, and dietary restriction seems to be trying to bring them closer to how the young animal looks. So our our focus right now is to be able to find good markers for each of these clusters, using which we can isolate those specific markers of intestinal stem cells, grow them in culture, and see which population or cluster is responsible for that enteroid formation. Is dietary restriction keeping them, a certain cohort of this ISCs, in a poised state such that they are ready for proliferation and differentiation? And if aging actually keeps them more quiescent, and they lose their proliferative capacity. And once this is established, we want to do the paired transcriptomic and methylome studies in single cells, which would be much more meaningful when we look at the methylation changes and the gene expression changes. Then the other aspect is, as I mentioned earlier, the transcriptome and methylome studies are usually association studies. So now that we have this enteroid model system that we can use and a functional readout that you can actually visualize, we are currently working on trying to bring about a cause and effect with these methylation changes. So the first thing that we are trying to do is the 38 genes that we found that changes with age and are reversed with dietary restriction. Using the CRISPR-Cas, we are trying to knock those genes out from the intestinal stem cells, either a single gene or combination of genes, knock them out and see if it has an impact on enteroid formation, and figure out which are the genes which are actually responsible or required for that enteroid formation. Now, the data that I'm showing here, this is actually not mouse enteroid. This is actually human enteroid. In collaboration with a surgeon here, we were able to get some intestinal biopsies, and from those we were able to get the crypts and grow them in culture. And this is, which is ongoing, so which is fresh out of the lab right now. And we were able to transduce that with a CRISPR-Cas, with a guide RNA for one of the genes that we are interested in. And this basically shows the transduction efficiency with GFP. And we are knocking it out, the gene, and then we are following it for enteroid formation, just to see if that particular gene is important for enteroid formation or not. So once successful, once the experiment is tweaked with all of this, our next step would be to transduce it with CRISPR-Cas9, but this time use a DCAS and fuse it with proteins which are responsible for, or enzymes which are responsible for DNA methylation and demethylation, basically tether them with DNMTs and TETs, and see if we can epigenetically modify each of those CPG sites that we have, and see if that has an impact on gene expression. And if it has an impact on gene expression, and that has an impact on enteroid formation or not. So this is where we are at, still in the beginning stage, long way to go. And with that, I would like to acknowledge my team, my bioinformatics person, my senior research associate, and my grad student who's been very helpful in getting each of those data, my collaborators, and all my other funding agencies. And thanks once again for giving me this opportunity to present my work.
Thank you, Dr. Unnikrishnan, for that really interesting talk. And it is so promising to hear about this metabolic memory, especially for those of us that are involved in the translational end of, getting to humans. It is very, hard pill to digest, if you will, to, have these lifestyle interventions be followed for a very long time. So we look forward to those biomarkers that you are exploring, and we will get right to some of the questions. Thank you again. Chris Wiley, Dr. Chris Wiley says, awesome talk, Arch. He's using what you'd like us to call you by. And then his question is, what are your mechanistic thoughts on how methylation is lost during short-term dietary restriction? Basically he's saying, such as lower levels of methyl donors in their diet or produced by aging-associated bacteria.
So that's a very good question. So this particular question has different levels. And the short answer to that question is we do not know right now. But the angle or the direction that we are taking is yes, if there are changes in the SAM levels, so we have to measure the donor, universal donor level in the intestinal stem cell itself, whether that changes with short-term diet, with age, and then with short-term dietary restriction, if there is a change in that. And also, how are these targeted to those specific sites? That's another big question that we have. It's a challenge as far as epigenetic studies or DNA methylation studies are concerned. Why those CPG sites and why not others, right? So those type of questions are something that we are hoping that we would be able to answer in the near future. But to your question of reduction in methylation, yes, reduction in SAM levels could be responsible, but we also do see hypermethylation, methylated regions with the short-term dietary restrictions. So it varies.
Yeah, thank you. We did have a question in the earlier part where you did talk about weaning and, later feeding. Dr. Larissa Constantino asks, how would dietary restriction affect reproduction? Are there life stages in which dietary restriction is most beneficial and stages of life in which it may be potentially detrimental?
So these are studies that I have not done, but people have done it in rodent models. Calorie restriction does reduce a little bit of the reproduction capacity, but what happens is they improve the ovarian aging in the sense that the reserve follicular cells and everything, the number increases, and their reproductive age also increases. So for a C57 black 6 mice, if you start them to use them as a breeder at about 6 weeks or 8 weeks, so they are good. They can be used for reproduction until they are about 10 months or 11 months of age. Now, with calorie restriction, that can be extended further. So it goes beyond 10 or 12 months of age, but their number of litters, number of pups, and everything that might have an impact. So ovarian aging or reproductive capacity is good in sense that it has a reserve, but then actually that getting translated into the number of pups being born, not so great with calorie restriction.
Thank you. Dr. Shandong Wang says, great presentation, thank you. And his question asks, I was wondering if the short-term DR altered DNA methylation affects any gene expressions involved in cellular senescence, proliferation, or differentiation. If yes, is there any difference between intestinal stem cells and non-stem cells in older animals or between male and female animals?
So good question again. So yes, it does have an impact on senescence, genes related to senescence, proliferation, and differentiation. As far as gene expression is concerned, we haven't looked at methylation patterns in those genes, which is something that it's in our to-do list, and we will be looking at that. And all that work that we have done is right now only on the intestinal stem cells, but we are trying to expand out to look at other. Now, this is an endodermal based stem cell. We want to look at some which were mesenchymal stem cell based. So we are looking at hematopoietic stem cells and some other stem cell types to see if we are seeing similar changes with age and short-term dietary restrictions. So we don't know as of now, but that's something that we might have an answer to in the next few years.
Great, thank you. Yeah, and some of your data with the older animals is sort of promising as well. Yeah. Dr. Wiley has a follow-up question. He says also your purine pyrimidine transporters are fascinating, and of course, proliferation requires nucleotides. Did you happen to measure intercellular levels of these in your models? Intracellular, I think is good. Yeah.
Not yet, not yet. So, like I said, we want to go across the biological spectrum. So, we are picking it one by one. So, this is all in the works. So, we would be looking at those transporters. And then we are also waiting to see out of the gene set that we saw, which is the one which is going to show the most effect as far as enteroid formation is concerned. And then, based off of that, we would be doing further protein analysis, looking at activities for those, and then even the metabolomics. Our hope one day, as you mentioned, Dr. Das, earlier, is to come up with a biological marker across the spectrum, a methylome marker, a transcriptome marker, a protein and a metabolome marker, which can be used, or we can create an index of that. I mean, we'll have to find that out from each of these different tissues, and how much of that can be cross-referenced to the plasma levels, and which is the one thing that we can actually measure in humans to see if we can create an index of biomarker which can be used at a translatable level. So that's the hope.
Yeah, thank you. And we write out that hope as well.
Yeah.
That pretty much is all of the questions that we had. Of course, this was a great discussion in the form of questions. Since we have a few minutes, I have a quick question with regards to any thoughts that you could share with regards to the GLP-1 receptor agonistic work that you're doing, because as you know, that's a favorite hot topic right now.
So, the GLP-1 study, the reason why I started the GLP-1 study was, the thought process was GLP-1 receptor agonist could be a DR mimetic, can be a calorie restriction mimetic, because all of those we see that in the humans where we have all the cases where it's not just reduction in body weight and fat mass. I mean, there's reduction in food intake. And even in rodent models, the treatment with GLP-1 receptor agonist, there is across the board, they were saying there was reduced food intake. But all of these studies were done on unhealthy models. They were either done on diabetic models or they were done on obese models, and they were able to see a reduction in food intake, which correlated very nicely to their changes in body weight as well. So what we have, so hoping that, okay, this can be a DR mimetic, we started out treating we used the Het3, which is the four-way cross created by Rich Miller's group at University of Michigan. And we started treating them with exenatide, which is a GLP-1 receptor agonist, both the young as well as old. So we had a 2-month-old, and then we started it at about 20 months of age. So we started at 2 age sets. And what we found was, in the old animals, the GLP-1 receptor agonist reduces body weight. And with the Het3, you can actually see that there is variation. So even if they are from 1 litter, the body weight is very different from each other because they are genetically more heterogeneous. So you can see. And when we give them this GLP-1 receptor agonist treatment, you can see the ones which had the highest weight is the one which actually is showing reduction, most reduction. So there is reduction in body weight as far as the old animals are concerned, but we did not see any change in food intake. So which was very surprising for us. And with the young animals, we did not see a change in food intake. We did not see a change in body weight as well. So this was at a dosage that was what was predominantly used by most of the rodent model studies that is out there. So what we did was we increased the dose in the young animals to see if that has an impact. And that when we increase the dose, yes, we do see a reduction in body weight in the young animals, but again, no change in food intake. So which is also the reason why I'm not showing any of those data here, because I really do not know what is going on with those animal model right now. It doesn't seem to be at least in the healthy animals, which are not diabetic or obese, they don't seem to be, the food intake reduction is not happening. So we are trying to look at activity, if there is more locomotor activity, or if there's more thermogenesis, or if there is any uncoupling of mitochondrial respiration or whatever. So those are different kinds of things that we are currently getting into to answer that question. But like I said, in the old animals, immediately with the same dosage, we were able to see a reduction in body weight, but no change in food intake. And I think at this point, dietary, rather than GLP-1 working like as a DR mimetic, I think DR works through GLP-1. So the feeding and fasting that we have, that we see with the dietary restriction feeding, when we feed the animals, it's like we feed them just at when the dark cycle light is turned off, and within 1 hour they finish eating. So they are on a long period of fasting there. So that feeding and fasting might be changing their gastric emptying capacity, and that might be promoting GLP-1 secretion, and DR could be actually, and we do have some data to show that with short-term dietary restriction, the GLP-1 secretion, the plasma levels of GLP-1s in the old and the old DR, old goes down and old ad-lib goes down and old DR seems to be going up. And even in the intestine, we seem to be seeing changes in the expression of the receptor. So DR seems to be increasing the expression of those receptors. So those are just preliminary work that we have. So my thought process has changed from GLP-1 receptor agonist being a DR mimetic to DR actually might be working through GLP-1.
That's great. Yeah, this is why we do the science, right? Thank you. And it would be important to explore what the nature of that weight change is too. Older
Exactly.
mice are responsive, but what is it? So we are now at the top of the hour. Thank you so much again for joining us today and for sharing all your work and thoughts. And for everyone that's attending, thank you for listening and for the great discussion.
Thank you all again. Bye.